Hello, welcome you all to the family of BioGoogle. In this video, we will discuss about the Watson and Crick model of the DNA. Okay, you all know that DNA is nothing but deoxyribonucleic acid, and it's one of the most commonly available genetic material in all the organisms, either plants or animals or bacteria or most of the viruses. DNA is the major or predominant genetic material. Okay, but before Watson and Crick proposed the double helical structure of the DNA, they have taken the evidences from many discoveries. They have taken the evidences from many discoveries made by many of the scientists. Not only these many, they have taken with the help of along with this, many more scientists have contribution towards the double helical structure of the DNA. Okay, we will briefly observe what is the contribution of these scientists while proposing the double helical structure of the DNA by Watson and Crick. Okay, in the first or second class of this chapter, we have discussed that Frederick Mischer was the scientist who observed the pus cells as well as the fish sperm, and he named that substance into a nuclein. He named that substance as a nuclein. Okay, named the substance as nuclein. And later on, Richard Altman, he has renamed nuclein into nucleic acid. Nucleic acid. Okay, then later on, maybe it's in the 1869, in the year of 1889. Later on, these two scientists, mainly Rosalind Franklin and Maurice Wilkins, they have worked on X-rays. Okay, Rosalind Franklin on X-ray diffraction, and Maurice Wilkins on X-ray crystallography, and at the same time Erwin Chargaff has proposed Chargaff's rules. I think I have explained this in previous videos. Better I will add the link of this video in this description of this video. Better you go and click on that. Okay, I will add the link of Chargaff's rules. It may be a 5-mark question in your examination, so I will add a link of this video into the description of this video. Please click on the description and go for that video. That is explaining about Chargaff's rules. Just nothing but arrangement of the nitrogen bases or a complementary base pairing. Okay, so by taking the help of all these, the two major scientists, that is James Watson and Francis Crick, have proposed in the year of 1953. They have proposed the model of the DNA that is known as double helical structure of DNA. Double helical structure of DNA. Okay, they have proposed the double helical structure of DNA, which is accepted by all of the scientists. Okay, let's now go to the structure of the DNA proposed by Watson and Crick. The model is known as a double helical model, double helical structure of the DNA. Okay, so these scientists may help you in NEET examination. What is the contribution of these scientists? Okay, you should be knowing about all this. Better just make a list out of this in your book. Alright, so we'll directly move on to the double helical structure of the DNA, or salient features of Watson and Crick model of DNA. Salient features of Watson and Crick model of DNA. Okay. Before that, I am suggesting you, if you are visiting our channel for the first time, please press on that red colored bell icon, red colored subscribe button, and after that just click on that bell icon so that you will get notification whenever I post my next lecture or next video. Okay, so now we'll see the salient features. Salient features of double helical model of DNA. Okay, salient features means what are, how the DNA structure is. Firstly, the DNA is a double helix, or DNA contains is made of two polynucleotide chains. Second point means, firstly, it is made of two polynucleotide chains. We have discussed about the nucleotide, polynucleotide in previous classes. So, our DNA, the Watson and Crick model, is made of two polynucleotide chains. Okay. Next, the backbone. Backbone is made of sugar and phosphate. Backbone is made of sugar and phosphate. Backbone is made of sugar and phosphate. And nitrogen bases project inside and are perpendicular to backbone. Now we'll see how this can be shown structurally. There are two polynucleotide chains. Look at the diagram very carefully. Most of the people make mistakes in drawing the diagram itself. Okay, so drawing the DNA is a skill. I don't say I'm a skilled drawer or a diagram artist, but just I can say drawing the diagram is a skill. So look at the diagram very carefully. So, two polynucleotide chains, backbone made of sugar and phosphate, means this one backbone, two backbones are made of sugar and phosphates. For example, I can show sugar, and outwards it is a phosphate. So ester bond. So this is one backbone. Similarly, other will be on either side. So third one, nitrogen bases project inside and perpendicular to the bases. So nitrogen bases will be this side: adenine, guanine, cytosine, guanine. They are perpendicular to the backbone. Backbone is like this. This is perpendicular. Guanine perpendicular. So backbone is made of sugar, phosphate, which is turning outside. But the nitrogen bases are arranged perpendicular to the backbone, and they are facing inside. Sugar is at the middle, phosphate is at the outside, and the nitrogen bases are at the inner side. Nitrogen bases are at the inner side. Okay, next point. The anti-parallel polarity. Okay, the two strands show anti-parallel polarity. The two strands show anti-parallel polarity. If it is one strand, other strand will be on the other side. So for example, I am labeling this as the 5' end. I am labeling the other end as 3' end. Of course, the other must be 3' end. The other must be 5' end. What is this 3', 5'? Okay, we all know that the meaning of nucleotide is sugar, phosphate, and one nitrogen base. Sugar, phosphate, and one nitrogen base. So here we have a phosphate again, right? Phosphate again. So sugar, phosphate, and nitrogen base. This will make one nucleotide. This is one nucleotide having pentose sugar, nitrogen base, and phosphate group. And this is also one nucleotide. Like that, 1, 2, 3, 4, 5. In this strand, there are 5 nucleotides. Okay, we need to discuss about the ends. In this end, this nucleotide has a free phosphate group at the 5th carbon. When we draw a diagram of pentose sugar, the 5th carbon, 1, 2, 3, 4, 5. So the 5th carbon will be having a free phosphate group in this nucleotide. The 5th carbon will be having a free phosphate group. Hence, this end is known as the 5' end. This end is known as a 5' end. Okay. What about this? When we say this as the last nucleotide, first, second, third, fourth, fifth. Okay. In this nucleotide, the third carbon has free OH group or free hydroxyl group. So this end is known as the 3' end. Means at the first nucleotide, the 5th carbon has free phosphate group. So it is a 5' end. At the other end, this nucleotide has free OH group at the third carbon. First, second, third carbon. So it is named as a 3' end. The other ones will be, of course, opposite. Just because the other strand will be reverse. Other strand will be anti-parallel. I'm telling you, both are parallel, but one is running in this direction, other will be running in opposite direction. If it is a 5' to 3', this will be of course a 3' to 5'. Of course, a 3' to 5'. So they are having anti-parallel polarity. Okay. Next. The next important property is complementary base pairing. Next property is complementary base pairing. Again, remember Chargaff's rule. Complementary base pairing means, you all discussed about the Jargaf's rules. Means we have 4 nitrogen bases. Among the 4 nitrogen bases, the adenine always binds with a thymine by forming 2 hydrogen bonds, and guanine always makes 3 hydrogen bonds with a cytosine. Adenine forms 2 hydrogen bonds with thymine. Adenine, thymine will form 2 hydrogen bonds. Guanine, cytosine will form 3 hydrogen bonds. Okay, so this will make one base pair. This will make one base pair. For example, if you want to show this here, if I have adenine here, if I have guanine here, so they will make 2 hydrogen bonds. If I have guanine here, if I have cytosine here, they will make 3 hydrogen bonds. Similarly, cytosine, guanine, adenine, thymine, thymine, adenine, cytosine, guanine, guanine, cytosine, guanine, cytosine, thymine, adenine, cytosine, guanine, cytosine, thymine, adenine. So bonds, 3 hydrogen bonds, adenine, thymine, 2, thymine, adenine, 2, 3, 2, 3, 2, like that. So every nitrogen base will form 2 adenine bonds with thymine, 3 with the cytosine, 3 with the guanine, 3 with the cytosine, 2 with the, like that. The opposite strand also has the complementary base pair. The opposite strand also has a complementary base pair. Hence, the 2 strands will be joined very tightly just because of the hydrogen bonds. Okay, this is the next point. So this will make a base pairing. Base pairing. Okay, you all know that. We'll just wind it up. Next very important point. 2 strands are coiled in right-handed manner. The 2 strands are coiled in a right-handed manner. Right-handed or clockwise. Right, means they are coiling in this manner. For example, I have anything paper, we'll just coil it. I have a ladder, will be coiling in clockwise manner, right-handed manner. So that is called right-handed manner or a clockwise manner. So earlier the 2 strands were like this, they were like this. But as of the DNA structure, the Watson and Crick model, they are coiled each other. They are coiled each other, so it looks like this. When they are not coiled, it looks like this. Adenine, 2 bonds, thymine, guanine, 3 bonds, cytosine. Like that, it looks like this. When it is linear, means without any coiling, straight. But in nature, in all plants, animals, higher organisms, it's in a coiled pattern of a right-handed manner. Right-handed manner. Right. Okay. Now, what about the helix? We can use the word helical here. Helical. Right-handed or helical pattern. What about the length of this? In one base pair or a total helix. Okay. So here, angle of rotation is 36 degree for one base pair and 360 degree for 10 base pair. BP means base pair. What is this base pair? Cytosine, guanine, 1 base pair. Thymine, adenine, 2 base pairs. Like that, this is 1 base pair, 2 base pair, 3 base pairs. So it will keep on counting. This is 1 base pair. Okay, this gap is 1 base pair. This distance or the angle of rotation is 36 degree for 1 base pair. But when we take 10 base pairs, the angle of rotation is 360 degree for 10 base pairs. Okay, means the straight lines, they keep on coiling in a clockwise or a helical pattern. The coiling means it must turn over. Okay, in that turning, the angle is 36 degree for 1 base pair and 360 degree for 10 base pairs. That will make 1 helix. That will make 1 complete helix. Okay, that's how. Then pitch of the helix. Pitch of the helix. Pitch of the helix is 34 angstrom. Pitch of the helix is 34 angstrom. What is that pitch of the helix? We said 360 degree, 10 base pairs. Okay, so from 1 base pair, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this 10 base pairs will cover the 1 helix. 1 helix. So the pitch of 1 helix is 34 angstrom or the length. Okay, next. While coiling each other, at some point it will leave very small distance. It will leave a very small distance or small gap. At some other point it will leave the larger gap. It will leave the larger gap. The smaller gap is known as a minor groove. Minor groove. The larger gap is known as a major groove. Minor groove and major groove. Okay, here the minor groove and the major groove have different angles of rotation. Okay, here we said 360 degree. In that 360 degree, 120 degrees will be covered by minor groove and 240 degrees will be covered by the major groove. 120 plus 240, it becomes 360. Right. Next, when we say pitch of the helix, so out of 34 angstrom, 12 angstrom will be covered by minor groove and other 22 angstrom will be covered by the major groove. 12 angstrom by minor groove and 22 angstrom by major groove. Like that, they will cover 1 helix. So this will make 1 helix. And this is known as a pitch of the helix. From this point, so till this point, it is 34 angstrom. That is nothing but pitch of the helix. Pitch means length of 1 helix. Right. Then it is coiling continuously, but it will not leave the diameter range. It will not leave the diameter, means it will coil in a same diameter. For example, anything wire running inside the pipe. Imagine a wire coiling inside the pipe, plastic pipe. So the wire can go only in the diameter of the pipe, it cannot come outside. Like that, imaginarily this will not go outside the length. This will not go outside the length. Better I will write this towards lower side. I can use the end. Okay, so this is called the diameter of the helix. This is called diameter of the helix. That is nothing but just 20 angstrom. This is diameter of helix, means the coiling will not go out of this diameter. And one more thing, they will always run with the same distance between them. So they are called parallel but anti-parallel. Parallel but in opposite direction. Right. This is, so the diameter will write next. Diameter of the helix is 20 angstrom. Diameter of the helix is 20 angstrom. Okay, like that. One more thing, this one base pair is like one plate, the another base pair is like another plate, third base pair is like one more plate. So each base pairs are stacked one above the other. Each base pairs are stacked one above the other. Every base pairs are stacked one above the other. Okay, this is called stacking. This is called, because of the stacking, the DNA will get more stability. Okay, so this will provide more stability to the DNA. Okay, stacking along with hydrogen bonds. Of course, the hydrogen bonds will provide stability for the DNA, just to bind the two strands so that it will become more stable. Imagine there is only one wire, it is not that strong, but when it binds with the other to make a thread or any string, that will be more stronger than when it is single. Like that, the hydrogen bonds will join the two strands to make it more stronger, more stable. And along with that hydrogen bonds, the stacking or arrangement of one base pair or the other will also provide more stability to the DNA, more stability to the DNA. Right. So this is the basic structure of the DNA or double helical model of the DNA. Okay. So remember the distances, remember the data. Okay, don't confuse among yourself. And also you can convert this into nanometer also. You can convert this into nanometer. For example, if I am writing, better I will make a separate table for this to avoid the confusion. So I have written here 34 angstrom. 34 angstrom. It means 3.4 nanometer. Okay, and the distance between one base pair is 3.4 angstrom. 3.4 angstrom equals to 0.34 nanometer. 0.34 nanometer. Like that you can convert 20 angstrom. 20 angstrom equals to 20 angstrom equals to nothing but 2 nanometer. 2 nanometer. Okay, in examination some people may get, or you may get the questions in the form of angstrom, or you may get in the form of nanometer. You should be knowing how to convert all these three. Okay, you should be knowing how to convert all these three. Just because 10 angstrom equals to 1 nanometer. It is just because 10 angstrom equals to 1 nanometer. Okay, don't confuse among yourself. Practice this properly. Practice the thing properly so that you will be able to answer every question regarding the length or pitch of this. Okay, coming to NEET point. In examination, by looking at this, they will give the number of base pairs. For example, the DNA is having 6 base pairs. The DNA is having 6 base pairs, and the gap between each base pair is some numbers. The gap between each base pair is some numbers, and find out the length of the particular DNA. Find out the length of the particular DNA. Okay, they will give number of base pairs, gap between the 2 base pairs, and they will ask how much is the distance of that particular DNA, or else they will give total length of the DNA, and they will give the number of base pairs, and they will ask what is the gap between the 2 base pairs. They will ask what is the gap between the 2 base pairs. Okay, better I will do one thing. I will add some questions in the comment box. I will add one question regarding this in the comment box, and not description, comment box. So click on the comment box. It will be a first comment at the top. So you will have a question regarding this. So try to answer that question. So look at the comment box soon after the completion of the video. You look at the comment box and you will get a problem on this. Right? Till that, we'll wait for the next class for the next concept. Okay, so maybe in the next class we will see the types of DNA. Okay, alphabetically there are DNAs like A DNA, B DNA, C DNA, D DNA, Z DNA, like that many types are there. Okay, just on this basis. Okay, only the numerical changes are there, or some DNAs will be turning left, right, clockwise, anti-clockwise. So on that basis, just based on this numericals, there are few more types of DNA. Whatever we are discussing, it is a B DNA. Remember, whatever we are studying in our text, NCERT text, it is a B DNA. But we'll see some other DNAs also with respect to all this. Nothing but we'll just make a complete table. In that table we'll just study how these numbers vary. Right? So this is how we can study the Watson and Crick model of a DNA. Remember, in examination, either they may ask the structure of the DNA or the salient features of the DNA. Okay, answer is not the same. When structure is asked, should draw this and explain this in the form of paragraph. Explain these points in the form of paragraph. But when the salient features of the double helical model of DNA are asked, you have to write only these points. Right? So I think this is enough for the class. I think if you are visiting our channel for the first time, I'm requesting you, please press that subscribe button, red colored subscribe button, and press on the bell icon. And if you like the topic, today's topic, please forward this topic to our video to the friends, or if you are a teacher, forward this to your students or to one of your colleagues, so that they can also start learning the things. Okay, if you find any more doubts in this chapter, please add a comment, right? Till that, keep learning. And if you want any more videos regarding any chapter, please add a comment so that I can be able to come back to you very soon with the particular video of that chapter what you have asked for. Okay, thank you for listening till now. I wish you all the very best for your classes, learning and the further examination. Thank you one and all. Jai Hind.